I appreciate the invitation. Good morning from Chicago, and good evening, I guess, to any of you who are in Russia. I often like to start my presentations with a picture that's local, and in fact, this one comes from the last year meeting when Ilya arranged an outing for several of us to the Caucasus Mountains in the south of Russia, which was great fun. And this is a picture of several of us standing at the top of a peak of some sort. They're a little too small to see, but if you look closely here, you can see that there's our driver here on the left, and Ilya and Fabian and Morgan and me. Misha doesn't show up anywhere. He thought we were nuts, and so he stayed behind and he took this picture, fortunately. So I want to tell you about our efforts to adapt brain-computer interfaces, in this case motor interfaces, not the somatosensory ones that Sleeman was just talking about, to activities in the real world. Now, obviously, I don't mean mountain climbing, but what I do mean is using your hands for the kinds of activities that we might do, or that a spinal cord injured patient might do throughout the day in trying to do the sorts of activities that you and I do naturally. This is a clip showing the basic approach that we are taking to effect movement. This is using what's called functional electrical stimulation, and this particular clip comes to me by a colleague at the Cleveland FES Center, which is one of the world leading institutions doing this kind of work. This patient has an injury at about C5, so that means that his hand is paralyzed, and it's insensate and he cannot move it, but he still is able to move the proximal part of his limb. In this case, there are sensors, both implanted EMG sensors on the shoulder muscles and a mechanical joystick sort of thing at the shoulder that sense the movements of his shoulder, so that as he lifts his shoulder, it causes a pattern, a pre-programmed pattern of electrical stimulation of electrodes implanted in the muscles of his forearm and hand that cause his hand to close, essentially like this. You'll see here he lifts his shoulder, and this causes his hand to close. The moment he drops his shoulder, this causes his hand to open. So this provides significantly improved function for patients like this. It's been implanted in about 300 patients, but it requires significant cognitive burden to literally use the same muscles to control the position of the hand in space as to open and close the hand. It's a single degree of freedom control, and it's entirely pre-programmed, so the patient can basically just turn it on and off. What we have worked on over the last number of years is a similar kind of system that's controlled not by physical movements of the shoulder, but by recordings from the brain, using the typical kinds of brain-machine interface approaches that many speakers have talked about so far. But instead of controlling the kinematics of a robot or the kinematics of a cursor on a computer screen, we actually control the contraction of individual muscles. And this is the way that experiment works. This diagram was made in a Nature publication a number of years ago, but it describes the essence of what we are doing, and I'll tell you then how we're extending that. We record from the monkey's brain, and then in this case, the hand area of the motor cortex. We use those recordings to make predictions of muscle activity, much like anyone else makes kinematic predictions. Then we use those time-varying predictions of multi-muscle activity to drive a multi-channel stimulator, which stimulates the muscles and causes them to contract. We do this in the presence of a peripheral nerve block, so this allows us to temporarily paralyze the monkey's hand, and during this period, we do these experiments. And this is a clip showing this sequence. The initial couple of reaches that the monkey makes are during this nerve block, at which point his hand is paralyzed. Although, as in the patient that I showed you, he can still move his shoulder and his elbow completely normally because the location of the peripheral nerve block is at the elbow. And this is what it looks like. So these first couple of attempts are when his hand is paralyzed. You can see he can't grasp the ball, although he can reach toward it. And presumably, the neurons active in the hand area of the cortex are active as they normally are, as he's trying to grasp. Now we've turned the prosthesis on, and you can see he can use his hand not by activating those muscles through the spinal cord, but in fact through the computer and the electrical stimulator. So this was a basic proof of principle that we did a number of years ago. It used a relatively small number of electrodes on muscles, and it was limited to a very small range of motor activities. This particular one we chose because it does represent a natural kind of grasping behavior that might well be useful to a patient, but it's rather limited in scope. So what I want to talk to you about for the rest of this lecture is not the details of this brain-controlled FES, but it's our subsequent experiments to look at how different kinds of motor behaviors are represented within the brain. And we're doing this now wirelessly within a large cage that the monkey can move about and perform a variety of unconstrained motor behaviors. This is a picture of the cage, and on the right are the number of people who have been involved in these experiments over the years and currently. And you see this cage here, which contains apparatuses on the left that allow the monkey to do the lab kinds of tasks that we're used to doing, power grasp, button pressing, and so forth. And this panel here allows the monkey to push a button and get a water reward, as in the cage. On this side of the cage, we have a proximity sensor, so we can tell where the monkey is within the cage. And one of the things we ask him to do is simply walk back and forth in the cage. And that's what's demonstrated here. What makes this particular behavior more interesting, in addition to the button pressing at the one end, is the power grasp that the monkey makes as he walks across those bars. We can also give the monkey treats to play with. Here he's foraging for food inside this paper bag, also filled with paper. And here we're simply handing the monkey treats that he can grasp using precision grasp. So you get a sense of the broad range of activities that the monkey does in this cage that we can record from wirelessly with an implant you can no longer see on his head, which is the BlackRock wireless transmitter, and also a separate transmitter that's housed in this backpack that streams the EMG recordings. This is simply a diagram of the whole system, the wireless recordings of cortex and EMG that I've talked about already, and we can substitute within that backpack a wireless stimulator to do our FES experiments. Although the experiments that I'm talking about today use a system to record EMG rather than stimulate muscles, but the basic design here is very similar. This figure shows the three kinds of behaviors that I want to talk about for the rest of the talk. Power grasp, as we would do in the lab, the crawling task, which I described, including the grasps that the monkey makes on these poles, and a precision grasp, which requires a different shape and a more complex control of muscle activity than does the power grasp. Below these pictures are shown the rasters recorded from electrode arrays implanted in the hand area, and also EMG signals coming from mostly forearm and intrinsic hand muscles. And this is a limited set of the muscles that we are able to record at any given time. What I want you to notice is each of these tasks causes activity in the brain and in the muscles, and it allows us to look at the relationship between the brain activity and the muscle activity and ask whether or not the mapping or the relationship between brain activity and muscle activity remains the same across these different motor behaviors in different contexts. This work that I'll be talking about subsequently is largely done by a postdoc currently in my lab, Xuan Ma. So I need to take just a little bit of a divergence here from the main theme to talk about something that's actually rather close to the talk that Misha gave, although I promise I will not be talking about any twirly kinds of things here. But I do want to talk about the concept of a high-dimensional neural state space, which is indicated here. In this case, these are recordings that are made as the monkey's doing what we refer to as a center-out task. He grabs the handle of that robot and simply makes movements in one of these 8 directions. These peri-event histograms show the modulation of 9 different neurons that I've chosen because they're representative from the larger set of roughly 100 neurons. These traces here show the trajectory. In this case, the monkey was moving up or away from him and to the right. What I want you to notice is a number of neurons do largely the same thing. And if you look at these two here in particular, these are the prototypical Georgopoulos-like neurons that Misha also showed us. They become active about 100 milliseconds before the onset of the movement, which is shown by this green dashed line. And the firing activity persists throughout the movement. And at the end of the movement, as the hand is in a different position, it has a slightly different firing rate than it did at the beginning. And these two are doing almost exactly the same thing. Here's a neuron that's doing almost exactly the opposite thing. So again, Misha showed us this. There are neurons with preferred directions pointing in different directions. And in this case, the movement was in the anti-preferred direction. Here's a neuron that came on a bit later. Here's a neuron that came on and stayed on for quite a long period of time. And here's a neuron that shut off, but then became more active. So what I'd like you to take from this set of images is, one, a bunch of neurons do essentially the same thing. And this gets at the idea of a low-dimensional representation of movement by redundant neurons. But then there are also groups of these neurons doing different things. So how can we think about that more effectively? This is a diagram that I borrowed from Mark Churchland, in which we describe this neural state space. In this case, it's simply a three-dimensional coordinate system, and each of the three axes represents the firing rate of one of three hypothetical neurons. So think of them as any three that you might pick from this group over here. Depending on the firing rate of these different neurons, the overall activity can be represented at some location within this three-dimensional space. So a rightward reach might be located here, while a leftward reach that's accompanied by different firing rates in these three neurons would be located in a different space over here. This is 3 dimensions. You can imagine, although I can't draw for you, a 9-dimensional space like this in which the location of the neural activity is determined by the firing of 9 neurons. But you don't need to stop there. You can think about the 100 neurons that we can record, making a 100-dimensional space. And of course, we can also think about the million-dimensional space that is represented by the full number of neurons in this part of the brain. This gives us some mathematical tools to look to see how the firing rate is represented at any given point in time, and also how it evolves over time. And I'll show you another example here that comes from real data, not cartoons. At the top here, and this actually is a recording from somatosensory cortex, although it doesn't actually matter. Here we're recording 40 neurons, 41, and we would periodically bump the monkey's hand in one of these 4 directions shown here by this key at the bottom. As the arm was bumped, we would count the number of action potentials, or the firing rate of action potentials, from each of these 40 neurons. So you can think about this as a location within this 40-dimensional space that I just described to you in the previous slide. And then we can ask, where does the activity move within this neural state space for these different bumps? Now, again, I can't plot that 40-dimensional space for you other than with these rasters, but I can use principal component analysis, which again, Misha described for us in the second talk of this session, to reduce the dimensionality of this 40-dimensional space. And I'm going to do that down to 2 dimensions. And when I do this, I get a plot like this. So this is analogous to that 3-dimensional cartoon that I showed you, but these are real data. These are the locations within this 40-dimensional space projected onto 2 primary axes. And you can see that the brain evolves or moves 2 clusters of points in these 4 different locations as the hand is bumped. This is just a static snapshot of the brain activity at the peak of the response to this perturbation. But as I said, we can also look at the time evolution of these signals. This is a movie clip, which is done exactly as this static picture is done here. Each of these dots, which I didn't say previously, represents the result of 1 perturbation. Here it's a static snapshot. Here I'm going to show you a clip of the evolution of this. So essentially I'm showing you about 100 perturbations at the same time. You'll get the idea looking here. Here, the brain state is just kind of randomly changing, leading up to the bump. Here comes the bump, and you see the brain state flying apart in these 4 different directions, but you can see the evolution of that brain state forming. So these are what I'm going to refer to now as latent signals. They're signals that we can extract from the firing rate of neurons, but in fact, we think that the latent signals are the fundamental component and that they ultimately cause the firing rate of these neurons that we record. And we can look at those and learn some interesting things about how different behaviors are represented in the brain. Now, this is another way of looking at the correlation between, in this case, different muscles, and I'll show you different neurons in just a moment. In fact, this is literally the pairwise correlations between a bunch of different muscles that we recorded during these different behaviors. If you look closely here, you see there's a cluster of rather well correlated neurons here, also here, and down here. This matrix and all of the other matrices were sorted according to a clustering algorithm that clustered these similar neurons together. And what I'd like you to notice is if we compare power grasp, and that's power grasp in the lab, and the power grasp the monkey does by grasping the rods, the patterns of muscle activity in this spatial sense look quite similar to one another. But if we compare either of these 2 power grasps to the precision grasp, the patterns of muscle activity are rather different because, as I told you, the precision grasp requires a different set of muscles to be active than does the power grasp. I can do the same thing for neurons, and now we see something rather interesting. This is exactly the same analysis, but now the power grasp in the brain is represented in very much the same way as the precision grasp in the brain. You see the repeated elements here and here in both matrices. There are some additional elements here that are present here not as strongly, but basically these 2 matrices look rather similar, and they both look completely different from the brain representation of the power grasp during crawling. So this tells us that the context in which this power grasp is produced appears to be quite important. Now we can quantify this. Instead of just looking at the similarity of these matrices, I can do a scatter plot of each of the elements of these 2 matrices, which is shown here. So this is a scatter plot of the similarity between the power grasp and the crawling in the brain. And you can see it's basically just a random relationship between these 2. If you calculate an R, it's just 0.23. If we do the same thing for the representation of power grasp and precision grasp in the brain, in fact, there's a pretty strong correlation. So this is consistent with what we thought we saw just by looking at the matrices. The 2 kinds of grasps, despite their different EMG patterns, are represented rather similarly in the brain. And the power grasp, when it's done in 2 different contexts, is represented quite differently. We can just provide numbers for all the combinations of comparisons between these tasks. And just to reiterate what I just said, in the brain, the 2 types of grasps, power and precision, are quite similar. In muscles, on the other hand, the power grasp and the crawling grasp are quite similar. So this suggests that there are important differences in the representation of these activities in the brain. And this is another way of looking at what I just said. I claimed there weren't going to be any twirlies, but this is something similar. These are the trajectories that are traced out, just like that bump movie I showed you, when the monkey executes these 5 different kinds of behaviors. 3 of them I've already described, but we also had the monkey picking these treats, and I showed you a video of that, and also executing a wrist movement task. So that you can see the brain state evolves in completely different parts of this state space for these different behaviors. So what implications do this have for our ability to do a brain-controlled FES prosthesis? Here are predictions of EMG activity based on our recordings from the brain for the 3 different behaviors that I've talked to you about. Each of these predictions was made by training and testing on the same behavior. They were cross-validated, which means we used held-out data for these test examples, but they were trained and tested on the same type of behavior. And you can see these predictions are quite good. If we calculated R-squared between them, they'd be in the range of 0.7 or 0.8. On the other hand, if we train on the power grass and then test on either crawling or precision grass, our predictions are much worse. The crawling behavior, which includes power grasping, can't be predicted at all. The predictions are terrible. The precision grasp, which I told you was closer to that of the power grasp, we can predict okay, but really not terribly well. This matrix on the right shows us a summary of these within and across EMG predictions. Along the diagonal shows the quality in variance accounted for, which I told you before were in the 0.7 to 0.8 range, for training and testing on the same behavior. The off-diagonal terms represent training and testing on different behaviors, which I showed you work, in fact, I'm showing you right now, work rather poorly. So this has really kind of dire implications, at least for the use of these linear types of decoders, in terms of decoders that will generalize across a broad range of different behaviors, unconstrained behaviors. So we're in the middle of developing much more complicated nonlinear decoders, which I'm not going to tell you about because we're in the midst of that. The take-home message is that, in fact, they work significantly better than linear decoders, but we have a significant distance to go in making them work perfectly well. So this is the summary of what I've told you. We can use cortical activity to predict EMG and to control FES intensity across a range of different behaviors, and it works quite well if we train and test within the same behavior. However, the latent spaces in the brain in which these behaviors are represented are quite different, causing our linear decoders to fail across those behaviors. And we're working now, as I just said, to develop decoders that are more robust across this range of behaviors. And these are funding sources and colleagues that helped with this research. Thanks.